The Australian Research Council Centre for Excellence for Innovations in Peptide and Protein Science is a national research centre that brings together experts from Australian universities, publicly funded research organisations, and other research institutions in Australia and overseas. We aim to discover and innovate peptides and proteins derived from Australian flora and fauna to strengthen research capabilities and address pressing global issues. Our researchers have access to cutting-edge technologies at their respective institutions that enable them to undertake high-quality and transformative research. Our work begins by unlocking the peptide universe contained within Australia's plant and animal species. This work is driven by our researchers at the University of Queensland's Institute for Molecular Bioscience, the University of Sydney's Australian Wildlife Genomics Group, and Edith Cowan University in Perth. At the University of Queensland Institute for Molecular Bioscience, we house the Clive and Vera Ramachardi Drugs in Plants facility and one of the largest venom libraries in the world.
The butterfly pea is a beautifully vibrant plant with a mesmerising blue flower. Aside from its stunning appearance, the plant contains a class of peptides called cyclotides that we've studied extensively to understand their many applications in medicine and agriculture.
We are researching the venom composition of key Australian species using state-of-the-art genomic, transcriptomic and proteomic approaches, as well as a wide array of functional assays to identify the bioactive components. Spider venom is known to be a complex mixture of neurotoxins that have evolved to paralyse prey. When we unscramble this mixture and pick out novel bioactive peptides, we can potentially transform them into new life-saving molecules.
We use large and powerful spectroscopic technologies, like an ultra-powerful magnifying glass, to look into the molecular universe and visualize the shapes of biomolecules. At the Institute for Molecular Bioscience, our researchers use the 900 MHz nuclear magnetic resonance spectrometer to reveal structural data on complex venom and plant molecules. In addition to unlocking genomic sequences within venom, our researchers use advanced genomic techniques to unveil hidden details in the DNA that will allow us to formulate a plan to protect Australian biodiversity, or to detect unwanted proteins in processed food products, like gluten in gluten-free food.
We work alongside conservation managers and different communities to collect DNA samples from Australian wildlife. Using these samples, we are building a library of genomic resources to help develop better conservation and management tools for protecting Australia's unique species.
At ECU, we use proteomics to study crops such as oats, lupins and barley. By employing state-of-the-art mass spectrometry techniques, we investigate the proteins present in the food we eat and the beverages we drink. We are uncovering the bioactive proteins that promote health, as well as the anti-nutritional proteins that cause intolerance and allergies.
Having found new peptides and proteins, our researchers utilize advanced biochemical, biophysical, and chemical techniques to determine their structures, discover how they can enter cells, and identify changes that enable them to reach a specific biological target and can be used as new medicines or eco-friendly pesticides.
Our research is at the interface of chemistry and biology. We use our interdisciplinary expertise to better understand how proteins interact with one another and control molecular mechanisms that sustain life. We use computational techniques to design new peptides and peptidomimetics that bind proteins, chemical synthesis to construct them, structural, biochemical and cellular methods like NMR spectroscopy, fluorescence polarization and microscopy, flow cytometry and cell signaling to elucidate biophysical and biochemical properties of peptides and their chemical mimics.
This is an exciting area in chemical biology, as there is a growing interest in developing peptides as biological tools to deliver drugs inside tumor cells to treat cancer, or across the blood-brain barrier to treat neurological diseases. At QT, We use biophysical techniques such as fluorescence spectroscopy and surface plasmon resonance to characterize how peptides interact with the lipid bilayer of cell membranes. And we use flow cytometry and mass spectrometry to quantify how much peptide and protein is getting inside cells.
In order to know if a peptide or protein is reaching its intended target, our researchers are developing cutting-edge sensors and probes to enable us to visualize these cellular interactions.
Molecular sensors are valuable tools for identifying biomolecules that regulate physiological or disease-related functions. We work at the interface of synthetic and biological chemistry, developing synthetic tools to detect phospholipids on the cell surface and tools to identify chemical changes that can regulate protein activity.
My lab develops fluorescent sensors that emit light to illuminate biochemical changes in the body caused by neurodegenerative diseases. This technology is vital for improving medical diagnoses because it will allow us to identify disease-causing factors and intervene in their progression.
We use spectroscopic technologies to closely examine the atomic surface of peptides and proteins. This will allow us to understand the shape and size of complex biomolecules and design machine learning algorithms to reprogram peptides and proteins as functional biomolecules.
Nuclear magnetic resonance, or NMR spectroscopy, is a powerful technology that provides an atomic view into the molecular universe. For the NMR experiment, the aqueous solution of peptide or protein is placed in a strong magnetic field. The information collected provides detailed structural data of the molecule.
Using this structural information and other physical or virtual databases, we can use machine learning algorithms developed in our lab to build informative and predictive models. These models can help us create new and unique peptides and proteins to advance the discovery of life-saving medications and safer pesticides.
After finding promising peptides, we need new methods to produce them at scale. To do this, we use a range of approaches, including electrifying peptides and proteins, using flow chemistry, or harnessing enzymes and engineering them to become expedient tools for complex designer peptide synthesis, with a wide range of industrial, agricultural, and medical applications.
In our lab, we perform electrosynthesis and cyclic voltammetry experiments using an electrosyn device, which is a state-of-the-art instrument with simple functionalities that allow us to transform peptides and proteins into functional bioactive molecules.
The pharmaceutical and biotechnology industries are invested in developing peptide and protein therapeutics that belong to a growing class of drugs called biologics. My lab is focused on developing new, faster, cheaper, and more sustainable methods to produce these high-value molecules. Recent examples from our Centre of Excellence include the use of microfluidics to produce semi-synthetic bioactive proteins and the chemical synthesis of cyclic peptides as inhibitors of proteins that are involved in the SARS-CoV-2 viral infection and COVID-19.
A key area of interest in our labs is to develop the next generation of antibiotics to overcome the global challenge of antimicrobial resistance. We also use a combination of cutting-edge structural biology techniques to elucidate the structures of protein-ligand complexes, enabling our understanding of how drugs work at the molecular level.
We are reprogramming the genetic code in tomato plants to produce beneficial peptides such as cycletides. Our approach transforms a simple tomato plant into a sophisticated biofactory that will produce peptides at scale. This blue sky research will allow us to efficiently and sustainably grow next-generation therapeutics in plants for medicinal and agricultural purposes.
Our Centre of Excellence aims to discover, innovate, and advance the field of peptide and protein science in Australia. We strive to innovate new chemical and biochemical methodologies to transform peptides and proteins into functional tools with applications in biodiversity and conservation, sustainable agricultural, farming, and future medicines.